we have studied the function of other regions of the tDNA, that what role it is playing. So tDNA, it is playing role in the synthesis of some phytohormones like auxin biosynthesis or cytokinin biosynthesis. और ये जो रीजन है tDNA का, इसको हमने नाम दिया था कि इसमें ऑन्कोजेनिक जीन्स होती हैं, ऐसी जीन्स जो कि अनकंट्रोल्ड सेल प्रोलिफरेशन में रोल प्ले करती हैं. तो इसकी क्या रीज़न है कि क्यों सेल की जो ग्रोथ है वो डिसऑर्गेनाइज्ड होती है या अनकंट्रोल्ड होती है, कि जो मल्टीप्लाई करते रहते हैं यहाँ तक एक ट्यूमरस ग्रोथ प्लांट में बन जाती है. तो ये वजह है ऑन्कोजीन्स की वजह से. और ऑन्कोजीन्स का जो प्रोडक्ट है वो है फाइटोहार्मोन्स लाइक ऑक्सिन्स एंड साइटोकाइनेंस. बहुत सारी म्यूटेशनल स्टडीज ने ये इंडिकेट किया है कि जब ट्यूमर में म्यूटेशंस हो जाती हैं या ऑन्कोजीन्स में म्यूटेशन आ जाएं तो उसकी वजह से जो ट्यूमर है वो एबनॉर्मल हो जाता है. इन द सेंस दैट समटाइम ट्यूमर्स दे मे बी वेरी लार्ज साइज और एबनॉर्मल, एंड समटाइम दे कैन शो shooty growth or Rooty phenotypes. And functional analysis have shown that Rooty phenotypes, it is because of the high production of cytokinins, and shooty phenotype of tumors, it is due to the auxin biosynthesis. So these two phytohormones, they are disturbed as a result of mutations in the oncogenes that can result in the abnormal phenotypes of the tumor. We have early studied that cytokinin and auxins with reference to plant tissue culture, they are very important hormones because their appropriate balance, it can result in the organogenesis from the callus, or you can say that new tissues can be formed when we keep the balance of these two phytohormones. When their balance, it is disturbed, then tumor morphology, it is also disturbed. So we can say that the one region of the tDNA, it contains genes that are involved in the phytohormone biosynthesis. Although there are some other genes whose function is not completely known, but at least some genes are there who are going to synthesize some important modified amino acids that are commonly called as opines. Now, opines mainly they are two types, octopines and nopalines. इनका क्या फंक्शन है कि ये एग्रोबैक्टीरियम को कुछ न्यूट्रिएंट प्रोवाइड करते हैं, लाइक मॉडिफाइड अमीनो एसिड्स या कार्बोहाइड्रेट, organic salts, or even phosphate salts, they can also provide to the agrobacterium. So they can use the host machinery to synthesize nutrients for agrobacterium tumefaciens. So in this case, the promoter of nopaline and octopine synthetases, they can be their promoter, they are very strong, and they can be used to get the high expression of other genes that have been transferred to the plants. Now we will focus that what type of genes this tDNA or transferred DNA in the TI plasmid it contains. So this table shows the functions of some tDNA genes in Agrobacterium tumefaciens TI plasmid. So first we can focus on these genes OCS and NOS. So these genes, they are synthesized octopine synthetase or nopaline synthetase that play a role in opine synthesis. In addition to that, there are some genes for auxin biosynthesis, that is very important plant hormones, and these genes, they may be named as IAAH or IAAM, synthesizing very important enzymes that can mediate the oxygen biosynthesis, and it is tryptophan 2 monooxygenase and indole acetamide hydrolase. And another enzyme for cytokinin biosynthesis, it is isopentyl transferase. So these are very important genes that are located in the tDNA. So in addition to that, some other genes that are also present that play a role in the opine biosynthesis. If you remember, opines, they may be of two types, octopine and nopalline, and they are the source of carbon, nitrogen, and sometimes bacteria can also get phosphates that is mediated by these genes.